We welcome our next keynote speaker, Branka Zukic, research professor at the Institute of Molecular Genetics and Genetic Engineering, University of Belgrade. Personalized medicine uses multidisciplinary approach and high-throughput technologies which enable the analysis of so-called omics to improve treating of diseases, tailoring therapy to an individual patient based on his genomic uniqueness. Branka, the stage is yours.
Thank you. Thank you all. I have a very difficult task to speak after Vesna and Ognjen, but I'm coming from a little bit different surroundings. I'm coming from academia. And I will show you what we can do in Serbia with big data and personalized medicine. I'm working at the Institute of Molecular Genetics and Genetic Engineering, University of Belgrade. We have in total a little bit than 150 researchers. We are doing on human molecular biology, plant molecular biology, microbial molecular biology, and our main focus is research, but we also provide a service, molecular testing diagnostics. I wanted to talk to you about the magical molecule of DNA. And now we can all say that all diseases or medical conditions, except of soft trauma, have a genetic component. And humans have about, it depends what kind of book are you reading, 20,000 or even more genes, but also some genes that are called regulatory molecules. But it's only about 3% of human DNA is genes. The rest, we used to call it junk, but now we know that this is the most important, also the important for the functioning of our systems. So DNA is information. The letters in the DNA are only 4, A, T, G, and C. You have heard of them. And they are organized into words, codons, codons into genes, genes into chromosomes, and the book of us is called genome. It contains about 3 billion base pairs. So we are all identical, 99.9%, but the rest of the DNA contributes to the difference between us. So this diversity affects both morphological and physiological characteristics of humans and those related to our health and disease. So we are coming to the personalized medicine, or 21st century medicine. It aims to individualize treatment for each patient, which means to give the patients the right drug at the right time. It is also called precision medicine because it uses omics. You heard before of the omics. So it's multi-omics based medicine. You can use genomics, epigenomics, transcriptomics, proteomics, and other omics, including radiomics, to personalize the treatment for the patient. Our group for molecular biomedicine at the Institute have several ways in research in personalized medicine. We are doing molecular oncology testing, comprehensive genomic profiling. We are reading the somatic DNA and try to identify biomarkers important for diagnosis, prognosis, and therapy, both tissue and liquid-based, and we follow the somatic mutation regarding malignant clone evolution. Our main research theme is pharmacogenomics, and that I'm going to talk from the next slide. And we're also involved in clinical studies, and all of these big data need NGS-related pipelines and omics, bioinformatics tools, and analysis. And also from this data, we use prediction and machine learning modeling to find the patterns of biomarkers that can't be easily seen from the sequences. So pharmacogenomics aims to determine how genetic profile of an individual determine his or her response to a certain drug in terms both of efficacy and toxicity towards achieved personalized therapy. So the same group of patients has the same prescription and the same dose, but some of them are responding correctly as we wish. Some of them didn't respond at all, and some have toxicity. These differences are written in those in our DNA. So the reason we could go further in our research is the project we got from European Union. It is oriented towards pharmacogenomics in the Western Balkan, and we are coordinating this project, and we have European partners from Patras, Greece, from Ljubljana, Slovenia, and from Trieste, Italy. We also have some Western Balkan partners from Bosnia, Croatia, Montenegro, and Northern Macedonia, and they are in charge for the collecting on samples and working on dissemination. So the goals of our studies to use high-throughput DNA sequencing of Western Balkan population and to identify specific drug pharmacogenomic marker pairs relevant for Western Balkan region, and to design pharmacogenomic Western Balkan panel of genes for preemptive testing. Furthermore, we are designing electronic pharmacogenomic assistant for Western Balkan as a tool for implementation of personalized drug recommendation based on state-of-the-art knowledge on pharmacogenetics, on gene-drug phenotype association, and this tool will contain models for health professionals, biomedical researchers, and also for visitors. At the end, not so much important for today's story, we use induced pluripotent stem model system, cell lines, to validate novel pharmacogenomic markers. We are doing it in-house at the Institute of Molecular Genetics and Genetic Engineering. We have sequencing machines. You heard about Illumina. We have 3 of them. And we can do whole genome sequencing. We have also a BGI technology, long-read technology, and also following Sanger sequencing, real-time PCR, so everything that you need. We have also support from Office for IT and e-Government Directory for use of NVIDIA platform, supercomputer for doing this bioinformatic analysis, and to store our big data in National Data Center at Kragujevac, because we don't have enough space to keep all this big data. And what is the most important, we have trained people to do that. So the workflow of the project, in short, is to collect the samples, to do whole genome sequencing and bioinformatic analysis, and then to perform population pharmacogenomic analysis of the Western Balkan region. Starting from human genome, machine do the sequencing of short reads, and then we read those differences, these variants, and try to give them clinical meaning. So, Illumina technology, sequencing you also heard in the first talk. So we start from genomic DNA, then share it into smaller pieces, prepare the library, ligate adapters and indexes, and then pull the library and put it into the machine, and on the flow cell the magic is happen. So all the sequencing is happening there. So machine is reading fluorescence signal and turn it into raw reads of DNA, and it is called FASTQ file. So a FASTQ file, this is just an example how it looks. It puts some identifier of the sequence, raw data, and the quality score of each base. Everything above 30 should be considered as okay for further analysis. So in the first graph, you can see something that is very good quality sequencing, but sometimes or often you have sequencing data that should be pre-processed before further analysis, because some reads are of low quality, and then you have to trim the bases, and then or if you have, but you always have overrepresent sequences because of the adapters, you have to cut them out using some tools like CutAdapt or FastP. The pipeline looks something like this. So you have the raw data as FASTQ files, then turn it into some other file, like which contains raw data plus mapping information. It's called SUM or binary version of SUM, like BAM. And then at the end, you have a variant call file, which contains only information of the differences of the variants in your patients. So to align each read to the reference human genome, you can use several algorithms, and you get sequence alignment map or SUM file or binary version of that is called BAM file, which contains information from raw sequence and also information on position mapping and mapping quality. So then when you get those puzzles of short read into reference genome, you call the variants, and only information about different variants is written down. So at the end, you have a file that looks like this. This is VCF or variant call file, which contains only information about those variants. So it's the position on the chromosome, it's the referent allele, alternative allele, some quality info data. But how to know what does it mean? Is this is every of these variant important for pharmacogenomics? You should we have help for that, for interpretation of the variants using the pharmacogenomic knowledge base. It's the most comprehensive database, free. You can use it free. It contains all information about primary pharmacogenomic literature. The knowledge is curated manually. And there are information there about annotation, aggregation, and integration, clinical interpretation, and clinical implementation, because the association that work on clinical implementation, like Consortium for Pharmacogenomic Implementation, they are involved in this base. So this base annotates pharmacogenomics-based drugs dosing guidelines published by the consortia that work on recommendation for pharmacogenomics. It's Clinical Pharmacogenomics Implementation Consortium, or CPIC, or Dutch Pharmacogenetics Working Group. Pharmacogenomics knowledge base also annotates drug labels containing pharmacogenetic information from the drugs that are approved by FDA or EMA. And what is specific for pharmacogenes is that star allele nomenclature. This is not very typical for other variants, but sometimes one star allele could have two or more variants together. So it is not straightforward analysis, so you have to to take care of that. So this could be monitored by PharmVar and CPIC recommendations. So at the end, we used PharmCat, that is pharmacogenomic clinical annotation tool that extracts all pharmacogenomic variants and generate the report. There are some levels of evidence. For some drugs, testing is required. For some drugs, testing is only recommended. For some drugs, it is written that there are some information, but not with enough evidence to recommend the testing. And for most drugs, the genetic testing is only informative. So this base also contains information of that, and this tool can give us the information about each drug. So when we use PharmCat tool, the input file is VCF, and then the pre-processing happens, and only pharmacogenomic variants are extracted, and then matcher calls only those variants, and so I don't see. Yes, they are phenotyped with the third model, and at the end, reporter gives you a report that you can be read by humans. The idea is to have a translational tool, and we already made a prototype of electronic pharmacogenetic assistant that contains 3 modules, as I said, and this assistant should give us the information on if patients with this genotype and this disease treated with certain drugs may have this effect. It will be used for efficiently browsing pharmacogenomic information, to locate pharmacogenomic variants in NGS sequence samples, and to easily interpret the results. So 2 main features I would like to point out: clinical annotations that you can give. If you have variant, drug, and disease, you got the right annotation, and you can have the dosing guidelines, because the haplotype pair and drug information could give you the effect and recommendation for drug dosage. So this is our modest team and our way to change the world through the data. So thank you so much for your attention.
Thank you, Branka, for this very amazing talk. Please don't go away yet. I have a certificate of appreciation for you, for your participation. We can now have a coffee break and meet back here at 4:45. Thank you very much.